welcome to the slideshow on transcription, one of the fundamental processes within the central dogma of molecular biology. Depicted here is that central dogma, the flow of information from the top of the screen to the bottom of the screen, whereby which information encoded in DNA is transcribed into messenger RNA and then later translated into proteins, which tend to have the ability to carry out very specific functions within the cell. This is how we define what a gene is. A gene is a segment of DNA which is transcribed and later converted into some sort of functional molecule, whether it be proteins or a functional RNA molecule. RNA has a chemistry that is very, very similar to DNA. Remember that RNA stands for ribonucleic acid, DNA stands for deoxyribonucleic acid. The chemistry of these two different nucleic acids is very similar. They share the same phosphate groups. The sugar is slightly different. In deoxyribonucleic acid, at this position of the carbon ring, there is no OH group, whereas in RNA containing sugars contained within RNA, there is an OH group here. RNA also uses 3 of the same 4 nitrogenous bases as DNA, A's, G's, and C's. It uses a different base in place of the T's, which is called uracil or U. The chemistry again is very similar between T's and U's. The main difference is at this position here. That is not a position that is involved in any hydrogen bonding, so the uracils will end up forming the exact same hydrogen bonding patterns as the T's that we saw in DNA structure and function. RNA is made in a chemical process that is, again, highly analogous to what we have seen for DNA. Remember that DNA was synthesized through the activity of an enzyme called DNA polymerase. RNA is also synthesized by a polymerase. It is referred to as RNA polymerase. It has the same directionality as DNA. New strands are grown in the 5' to 3' direction, where which new bases are added onto a growing chain using a DNA template strand to determine what the sequence of those bases will be via the same rules of complementarity we saw in DNA, where A's match with T's, G's match with C's, and in the case of RNA, when there is an A on the template DNA strand, a U is added in the RNA strand. And again, just like in DNA, the template strand has the opposite orientation as the RNA strand. Here's the 5' end of DNA, the 3' end of the DNA. And in the RNA, it is opposite. 5' is at the top, and the 3' is at the bottom, and that is where the chain is grown from. At the end of a single chemical reaction catalyzed by RNA polymerase, 1 nucleotide is added to the end of the growing chain. A nucleotide triphosphate is the starting material. It gets glued onto the end of the growing chain, has the same sort of linkage that DNA does, this so-called phosphodiester linkage, and one of the waste products of this, just like in DNA synthesis, is 2 phosphate groups stuck together, what's also known as inorganic pyrophosphate. Shown in this slide are 2 alternate representations of the process of transcription. Shown on the bottom is a single nucleotide being added to a growing strand of RNA, shown in red. The template DNA strand is shown in blue. The individual nucleotides are shown as little kind of blue or red boomerangs. You can see the nucleotide triphosphate that is incoming to be added to the free 3' hydroxyl of the RNA. It is a U, which will be placed opposite the A in the DNA strand. On the top panel, we see less chemical detail of what is going on, but we see it on more of a macro scale. So we see a piece of DNA showed as this blue double-stranded helix. two different genes are being transcribed at once, gene 1 and gene 2. They are actually being transcribed in two different directions. So gene 1 is the template for gene 1 is this darker blue of the two strands, whereas the template for gene 2 is the lighter blue of these two strands. So as a result, the RNA polymerases, shown as pink blobs, are going to be moving in opposite directions with respect to each other, because one is transcribing the strand in one direction. Right? Here's the free 3' end of the light blue strand. Here's the free 3' end of the dark blue strand. So the light blue strand is going to be transcribed from right to left on the screen as shown, whereas, sorry, that was the dark blue strand. Whereas the light blue strand, shown here, is going to be transcribed from left to right. You can see what's often referred to as the transcription bubble, this small region of DNA that is being accessed by the RNA polymerase that is unwound. And during the process of transcription, within the bubble, the two strands are separated from each other. The bubble rewinds behind the RNA polymerase as it moves along the DNA, and it unwinds in front or upstream of the RNA polymerase. And there's a region of what's often referred to as a DNA-RNA hybrid helix that is a few nucleotides long within that transcription bubble. And the remaining and the rest of the RNA that's nascent, still being transcribed RNA molecule is spooled out of this complex as transcription progresses. So just like in replication, there is a fairly easy way to kind of mentally organize these various biomolecular syntheses. You can kind of think of them as kind of a story or any other process which has a beginning, a middle, and an end. And one of the nice things about central dogma processes is we are consistent in the terminology. We always refer to the beginning of the process as an initiation, the middle of the process typically as elongation, as the biopolymer is lengthened longer and longer, and eventually termination, where the process is ended in an organized way. So these three different steps of transcription, initiation, elongation, and termination, are characterized by key things. In initiation, we have a region of DNA that is referred to as the promoter that is recognized by transcription factors. These are proteins which will bind to the DNA at the site of the promoter. And these proteins, transcription factors, are going to enable the enzyme, RNA polymerase, to be able to bind, be recruited to the gene that we are trying to transcribe and form a complex that will allow initiation of transcription to kind of finish. Part of initiation is you have the DNA is unwound or pulled apart or melted so that the two strands are separate from each other. And this is an important step within the initiation. And it's often referred to as the open complex. So the end of initiation in bacteria is when you have several nucleotides have been transcribed, you have an initiation, the initiation factors have been shed, and the RNA polymerase then pauses and releases some of its components before it moves into a much more rapid phase of transcription known as elongation. Elongation essentially is the repetition of that key chemical reaction over and over again, whereas a single nucleotide is added to the growing RNA chain, and the nature of that nucleotide is determined by the template DNA. Once elongation proceeds and the entire gene is transcribed, a region of DNA will be recognized, which will initiate termination. And the termination signal essentially causes this complex of DNA, RNA, and the protein enzyme RNA polymerase to all dissociate from each other, and the complex is blown apart and they all separate, and that is the end of transcription. So we'll spend a bit of time looking at the first phase of transcription, the initiation phase, in a bit more detail. So the basic idea is that RNA polymerase is actually an enzyme which is a conglomeration of several different proteins. So it is a complex of proteins. And when they are all present together, it is referred to as a holoenzyme. So all of the pieces are there together. This holoenzyme will bind to DNA in random locations, and then it'll begin to move along the DNA or scan the DNA until it encounters a promoter region. When it does so, the holoenzyme will stop moving, it will pause, and then it will melt the DNA. Part of the key component of the RNA polymerase holoenzyme that recognizes the promoter is something that's referred to as the sigma factor. It's one of the several proteins, and it recognizes the promoter. In particular, two regions that are very important are called the -35 and the -10 regions, which are upstream of the transcriptional start site. A bit more detail on promoters. Promoters are defined as DNA sequences that quote-unquote promote gene expression. Remember that gene expression is a term that talks about the ability to take information stored on DNA and convert it into a functional molecule. So for many genes, gene expression is the combination of both transcription and translation. If a gene encodes just a functional RNA, gene expression requires just transcription and no translation. And typically promoters are located just upstream of the site where the gene begins. So they are adjacent to the gene, and they are recognized by the RNA polymerase, and therefore it can correctly align itself with the beginning of transcription, or what's also known as the transcriptional start site. So here is a depiction of what a promoter would look like. If we use this numbering system, +1 is where transcription begins. So the +1 site is the first nucleotide that is actually transcribed. That information ends up in the RNA. As we've shown it here, we're showing both strands of the DNA, both the template strand and the coding strand. Only the template strand is used in making the RNA. So the RNA uses complementarity against the template strand only. The coding strand is not used. But because both the coding strand and the RNA are complementary to the template strand, they tend to have, well, they will have essentially the exact same sequence. The only difference being that the DNA will have T's and the RNA will have U's. The promoter is a region that is upstream of this start site. So if this is the start site, everything to the right of the start site in this particular figure that is shown gets transcribed. Everything to the left of it is the promoter. And we have two very key sequences. One is referred to as the -35 sequence. One is referred to as the -10 sequence, that are roughly 10 nucleotides and 35 nucleotides upstream of that transcriptional start site. There's obviously a spacer in between the -10 and the -35 region. We often don't pay a lot of attention to that because these sequences are conserved, meaning that there are very similar sequences found in essentially all the promoters of bacteria. So again, to give you a couple of balloons to remember things, bases preceding this +1 site are in the negative direction. So there is no base 0, there is a -1, and then directly adjacent to that is a +1. And the bases to the right in the positive direction, that is actually what is transcribed. And the -10 and the -35 sequence are sequence elements that play key roles in transcription. One of the terms that we use to describe the -10 sequence is the Pribnow box, which is named after the person who discovered it. What is shown here are now single-stranded sequences of DNA. We know it's DNA because it has T's instead of U's, of a bunch of different promoters all found in the same organism. They're all in E. coli. And they are arranged in a way that the -10 region and the -35 regions are highlighted. And when you align sequences like this, it allows you to see patterns of sequence similarity. So again, here's the transcriptional start site. This is the -10 region. This is the -35 region. And we can see that the -10 region in particular is highly conserved. The vast majority of the sequences share 5, sometimes all 7 of, 1, 2, 3, 4, 5, 6, all 6 of the nucleotides that are characteristic of the -10 region. The -35 region, there's a significant amount of conservation as well, albeit not to the same extent as the -10 region. So let us kind of take a break here and think about this conceptually. So my first question to you is, what is being shown above? What kind of molecule is being depicted above? And don't advance until the next slide until you kind of come up with an answer. So the molecule that was shown above is actually a DNA molecule. The key kind of things that you can recognize about this piece of DNA are that it has T's instead of U's, so we know that it's not RNA. It's all A's, G's, C's, and T's, and it's also double-stranded. So all of those things allow you to understand that this is a piece of DNA. The next conceptual question: what region of this DNA, region or regions, are most likely transcribed? So think about that and come up with an answer before you advance to the next slide. So in this depiction, here's our transcriptional start site, +1 region. Everything upstream of that transcriptional start site has negative numbering, -10 and -35. Everything downstream, +1, +2, +3, +4, +5, and so on, are the nucleotides that are transcribed. So region A is the only region of this DNA that is actually transcribed in this above in the picture shown here. All right. One more conceptual question: where is the promoter? Again, wait until you have an answer before you move on to the next slide. So the promoter is the combination of region B and region C, the -10 and the -35 sites. They comprise the promoter. And that's another clue that this is DNA, because this piece of the segment of a molecule contains the promoter, and promoters are not transcribed. You only find promoters on DNA. Another conceptual question for you to think about: which strand is the template strand versus the coding strand? So if you think about this, we have two strands. Our transcriptional start site here is at the +1 site, and this represents the 5' to 3' direction. Now transcription occurs in the 5' to 3' direction, which means that the actual template has to have opposite orientation to the transcript. So that means the top strand is the template strand, right? This is the 5' end, this is the 3' end. Of the top strand, the new RNA will be synthesized in this direction, in the 5' to 3' direction, so it has reverse directionality. The top strand is template, and therefore the bottom strand is the coding strand. So, to further take this question, we can infer, therefore, that the sequence of RNA will be exactly the same as the coding strand in the 5' to 3' direction, except that you replace all the T's with U's. So we would expect our RNA transcript to be A, C, U, A, U, G, G, C. And that's in the 5' to 3' direction. So a couple of key vocabulary terms that are associated with initiation of transcription in prokaryotes. When the RNA polymerase stops at the promoter, but before it melts the DNA, this is referred to as the closed complex. When the DNA is unwound in the promoter region, in particular the -10 site, also known as the TAT-AAT box, that's referred to as the open complex. Within the open complex, there's a short strand of RNA that is made, at which point the holoenzyme pauses and part of the holoenzyme is released. That's the sigma factor, and then transcription can continue, and that is the official endpoint of the initiation of transcription, after which the core enzyme is now free to slide down the DNA, continuing to transcribe, adding individual bases to the growing RNA strand until it finally reaches the termination step. If we look at this with pictures, we can see this represents the DNA strand. These different blobs or spheroids are all individual subunits of the RNA polymerase. There are 2 identical alpha subunits, 2 very similar beta subunits, beta and beta prime, an omega subunit, and then this sigma factor, which is about 70 kilodaltons in size. It's sometimes referred to as sigma 70, and it is what recognizes the -10 and the -35 regions of the promoter. At the end of initiation, you have a small amount of transcript that is made. You have an open complex. You can now see the 2 strands of DNA are separate from each other, and the sigma factor is shed, allowing RNA polymerase to enter the elongation step. This entire complex here is referred to as the holoenzyme. This is referred to as the core enzyme once you lose the sigma factor. An important thing to note is that there isn't primase activity required the way that replication requires primase. RNA polymerase is able to initiate synthesis de novo, meaning from the beginning, rather than elongating an existing nucleic acid that already is in place. So one more depiction of the initiation step of transcription, just from another textbook. You start with a piece of DNA. You have a very characteristic region highlighted in tan here, the promoter, that contains both the -10 and the -35 regions. The RNA polymerase will track along the DNA molecule until it encounters a promoter. The promoter will be recognized by the sigma subunit of the RNA polymerase, and it will pause and form the so-called closed complex. During this pause, the DNA will be unwound upstream of the transcriptional start site, and the RNA polymerase will begin to transcribe RNA. So there's a region of DNA that is unwound, what is often referred to as the transcription bubble, and in this case you have the open complex. After a small amount of RNA is transcribed, you get another pause whereby which the sigma factor is released from the complex, leaving only the core enzyme, which is then able to kind of proceed to the elongation step of transcription. One more depiction of this phenomenon from yet another textbook. In this case, we have our DNA, which is shown in green and pink. The green region is the promoter. The pink region is the coding sequence. RNA polymerase is shown as this kind of light tan spheroid. The sigma factor subunit is kind of shown as this hot dog shape. And you can see here's the formation of the closed complex, where it is the RNA polymerase holoenzyme, so the whole complex is recognized in the promoter and is stopped, but the DNA is not unwound yet. You get formation of the open complex in portion B of this, where DNA is now melted, and a single nucleotide is allowed to initiate transcription at the +1 site. RNA polymerase then will use its polymerase activity to add nucleotides in the 5' to 3' direction. So you can see the formation of a new RNA strand. There'll be a short region of it, which is in double helical formation with the DNA. That's a so-called RNA-DNA hybrid helix. And as the RNA polymerase moves from left to right on the screen, this will spool out. After several nucleotides, we have a pause of transcription. The sigma 70 subunit is shed because it no longer is needed to recognize the open complex. Sorry, no longer needed to recognize the promoter. The polymerase has moved beyond the promoter and is now actively transcribing the gene. That represents the end of the initiation of transcription, and then you enter the elongation step where the polymerase continues to add nucleotides and move from left to right on the screen. Here's yet another depiction of the transcriptional process. This depiction includes information that we know about what the polymerase and the DNA in the complex actually look like from structural studies. So what's shown here is this brown kind of blob. To a lot of people, it reminds them of a hand. This is the thumb of the hand, and these are the fingers. It is essentially grabbing the DNA. The DNA shown here is this double helix. And so this kind of hand blob represents the RNA polymerase core enzyme. And where it interacts with DNA, it actually kind of bends the DNA. And this bending of the DNA is one of the forces that contributes to the ability to separate the DNA and form this open complex. There's a channel that allows entry of RNA nucleotides or monomers. They're in the triphosphate form as they get added to the growing chain, and the nascent RNA that is being synthesized trails out the back. A couple things to kind of keep in mind as you think about this kind of a little bit more realistic picture of what's going on, but still simplified, is that this RNA polymerase is sliding along the DNA in this direction. The open complex, this bubble, is moving along with it as it moves along, and the RNA molecule keeps getting longer and longer. Here, the purple strand is the template strand. That's what's actually used to make the DNA, or the information in that DNA is what's used to actually synthesize the RNA. And just like for DNA polymerase, it is unidirectional. You're only making RNA in one direction, often referred to as the 5' to 3' direction. And that pyrophosphate release here is not shown in this image. So a lot of these things are very similar to the activity of DNA polymerase, but there are some important differences, such as U's versus T's. So that is the end of what we're going to talk about in terms of bacterial transcriptional initiation, and we'll move on to the subsequent steps of the process. So we go into elongation. What we saw for the open complex of initiation is still is very similar to what's going on during elongation, where you have an RNA polymerase shown here. There's a region of DNA that is unwound. There's a region of RNA that is hybridized, so it's forming those hydrogen bonds, G's to C's and A's to U's. The so-called RNA DNA hybrid region, and then you have the RNA that is tailed out, that is spooled out the back end. Right? And here we're looking at one of the things to keep in mind is, unlike DNA polymerase, you don't have the additional factors that are required to unwind the DNA in front of the polymerase machinery, so it doesn't require a separate helicase. And you also have rewinding of the DNA behind the disrupted area, the bubble caused by the RNA polymerase. So as a result, you don't get supercoiling and the need for gyrases and topoisomerases. So I'll include this slide because it has defined some of the terms in words that we've kind of already covered in the narration. If we look at the process in terms of numbers, the open complex, or that bubble, is about 17 bases long. That's the region of DNA that is not wound. It is unwound in front of the transcription bubble, and it is rewound behind the transcription bubble. And on average, in bacteria, RNA is synthesized about 43 nucleotides per second. So think about how fast that actually is. When we think about termination of transcription in bacteria, there are two main mechanisms that allow that DNA RNA hybrid to be disrupted. So essentially, if you can pull apart that DNA RNA hybrid helical region, double helical region, that will end termination. So one of these processes requires a protein known as Rho. So we often refer to that as Rho-dependent termination. And a second process doesn't require any proteinaceous cofactor, and that's called Rho-independent termination. So not particularly creative names, but they're very descriptive. So this is an example of a termination site for Rho-independent termination, and it has some key features. One is you can see that it tends to form a stem loop structure, and in particular this stem loop structure is heavily populated by G's and C's. Right? So almost all of these base pairs are GC base pairs as opposed to AU base pairs. And as you can see by the three lines that are shown in a GC base pair, we know that those form 3 hydrogen bonds and are relatively strong compared to AU base pairing. So this is a very stable stem loop structure because it has such strong hydrogen bonding. And downstream of this is a region that has a repeat of a bunch of U's. So why this is important is, you can show here this intrinsic mechanism, also known as the Rho-independent termination. As the RNA is spooled out of the RNA polymerase, that stem loop structure is formed. We know that this snaps together. There's a lot of force involved in that snapping together because it is such a stable stem loop structure. Within the RNA polymerase open complex, you have AU base pairing exclusively. Right, so that's relatively weak. So this is weak. This is a weak force of attraction between these. This is a relatively strong force of attraction between these, and the formation of this loop disrupts the RNA DNA hybrid helix, and therefore disrupts transcription, leading to its termination. So here's another depiction of that Rho-independent or intrinsic termination mechanism. You have this U repeat region. You have a stem loop structure that is formed, and that disrupts the DNA RNA
hybrid region, leading to termination. In the row-dependent termination, a lot of the there are some similar elements formed. There is a stem loop structure that forms, but it is mediated by this protein row. Protein row, because its activity is that it disrupts a nucleic acid double helix. It is therefore referred to as a helicase because it is essentially disrupting the RNA DNA double helix. So as we mentioned, transcription is most extensively studied in prokaryotes, but we are going to talk about some of the key features that differentiate eukaryotic transcription as opposed to the prokaryotic process. But just to repeat, many of the basic features of transcription are the same. The things you have to worry about when you're thinking about eukaryotes, they tend to be larger organisms. There's higher levels of complexity. There are more than one cell involved, and the cells themselves have increasing degrees of complexity. You have separate organelles and compartments within a cell which don't exist in bacteria. And all of those things are addressed by very specific mechanisms. So to start out with, unlike prokaryotes where there's a single RNA polymerase gene, there are actually multiple RNA polymerase genes in eukaryotes. And they are named in the order that they were discovered: RNA polymerase 1, polymerase 2, and polymerase 3. The polymerase that we have the most familiarity with, that we'll see a lot in this course, is RNA polymerase 2. That is the polymerase which synthesizes all mRNAs, so messenger RNAs, which go on to encode proteins. It transcribes some other functional RNAs, such as the small nucleolar RNAs, and then the other RNA polymerases primarily transcribe functional RNAs. So pieces of the ribosomal RNAs, are transcribed by polymerases 1 and 3. Polymerase 3 also transcribed all of the transfer RNAs. If we compare the initiation of transcription between eukaryotes and prokaryotes, we see that there are some similarities and some differences. If we look at the transcriptional start site, in prokaryotes, the transcriptional start site, there's a region of the promoter that is approximately -10 to -35 region upstream. The equivalent to the -10 region in eukaryotes is often referred to, there's a sequence that's TATA, it's referred to as the TATA box. It is recognized by a protein called the TATA binding protein, TBD, as shown here. So the TATA binding protein in eukaryotes is essentially functionally equivalent to the sigma factor in prokaryotes. Right? It recognizes the key components of the promoter. Similarly, as initiation proceeds, eventually the TATA binding protein, shown here, is shed as the polymerase ends the initiation stage and then enters the elongation phase of transcription. The sigma factor is shed, the TATA binding protein is shed, as well as some other components of the polymerase. All right, there are other individual components. There's a higher degree of complexity in eukaryotes. There's various transcription factors that play a role that don't necessarily have equivalence in the prokaryotic system. And the RNA polymerase itself is highly regulated. What's shown here is this C-terminal domain of the RNA polymerase with a bunch of P's on it, and these represent phosphates, and the activity of the polymerase can be regulated by hyper and hypophosphorylation of this C-terminal domain of the RNA polymerase. One of the other key features of eukaryotes that we have to reconcile is this multicellularity aspect. Right? We know that eukaryotic cells have a nucleus. The DNA resides within the nucleus, and this is therefore where transcription occurs. RNA therefore has to be exported out of the nucleus in order for the next step of the process to occur, of the central dogma processes to occur, which is translation. In prokaryotes, because there is no compartmentalization of the cell, a lot of times you have a case where an RNA polymerase is transcribing a gene, the nascent RNA is being translated before the RNA transcriptional process is even finished. So you oftentimes get a coupling of transcription and translation. It's one large kind of process. Because they're in different compartments in the eukaryotes, the RNA has to be exported out of the nucleus before it can ever be translated. Therefore, these processes are completely separate, and as opposed to being coupled, potentially coupled, in the prokaryotic system. Another key feature of the eukaryotic system is the RNA that is transcribed is typically untranslatable. It has to be processed before it can actually be translated, and the processing involves removing certain sequences of the RNA transcript, what are known as introns or intervening sequences, splicing back together the sequences that are shown in green, which are the exons, capping the RNA molecule on both ends. So at the 5' end there's a cap, a tail is added to the 3' end, that is a bunch of A's in a row, so that's often referred to as the poly-A tail. And these caps on the 5' end and the 3' end are necessary for the stability of this RNA so that it can be recognized by the ribosome and translated, but also be ignored by nucleases in the cell which degrade RNA. This slide depicts a form of RNA processing that is a separate process. This is not the processing of mRNA, but instead is processing required in order to generate the functional RNAs that are needed to construct a ribosome. So as we see here, we have a single promoter. It's driving the transcription of a bunch of different potential genes. They are transcribed as a single transcript. We'll learn more about transcripts that have more than one gene on them when we talk about gene regulation. This is often referred to as the 45S transcript. And the information for several different ribosomal RNAs are discrete regions within that large transcript. And in order for them to be freed up and allowed to form a ribosome along with a large number of auxiliary proteins, they need to be processed, and this is done by cleavage events that occur within the nucleus, and this is done in an area of the nucleus known as the nucleolus. So a lot of times you refer to the nucleolus as the birthplace of the ribosome, and that's because these cleavage events are occurring in that space. And at the end of processing, you get ribosomal RNAs of a couple of different sizes. Some are associated with the large and small subunits, and these have to be exported out of the nucleus into the cytoplasm so that they can be used to construct ribosomes. And again, these are the functional RNAs key in ribosome structure. So, in eukaryotes in particular, lots of different types of processing are required. And as we mentioned earlier, messenger RNAs require processing as well. The raw DNA looks like this. There's a promoter region. There is a sequence of RNA that gets transcribed. This is a depiction of what that raw RNA transcript, also known as a pre-mRNA because it is the precursor to mRNA. It is modified at the 3' end of that molecule by the addition of a poly-A tail. It is modified at the 5' end by the addition of a cap, both of which we will talk about in slight more detail later. And there are sequences within that, the so-called intervening sequences, also known as introns, which are removed through a process of RNA splicing. All of these, again, events occur prior to this now mature messenger RNA being ready to be exported out of the nucleus into the cytoplasm, where it can then be translated by ribosomes. So if we look at these three different processes, the capping is what's called a 7-methylguanosine cap. It's important for a lot of different processes that the RNA is involved in. One of them is export into the cytoplasm, recognition as part of the translational process, as well as the splicing process. So the 7-methylguanosine cap addition is one of the first steps in the maturation of that raw or immature RNA transcript. Here's the chemical structure of the 5' cap. This represents the 5' end of the messenger RNA. This material is the 7-methylguanosine, and you can see some additional chemical modifications are made to the first and or second nucleotides in the sequence. Poly-A tailing occurs at the opposite end of the messenger RNA, so it's at the 3' end. It is a sequence of A's that is not encoded in the gene sequence. Instead, they are enzymatically added to the messenger RNA after transcription has already terminated. So here is a visual depiction of the process of the addition of the poly-A tail. You can see here is the transcription bubble that occurs following transcriptional termination. The RNA strand is cleaved, so nucleotides that are downstream of a particular recognition sequence are removed. Then there's a gap of 15 to 30 nucleotides. This then becomes a substrate for an enzyme that's called poly-A polymerase, which will add a number of A's to the end of the mature messenger RNA, and the number of A's is highly dependent on species. And the third type of processing that occurs to create a messenger RNA is splicing. So the splicing is the removal of the intervening sequences, the so-called introns, and the rejoining of the exons that were interrupted by the introns. There are a couple of different ways, mechanisms that eukaryotes use to remove introns. Some of the mechanisms, such as group 1 and group 2 introns, don't require any sort of protein to catalyze the reaction to remove the intron. So the RNA itself has catalytic activity, and some of them all are mediated by proteinaceous entities such as the spliceosome. However, all three of these cases, the end result is very similar. You have the removal of the intervening sequence of RNA, the intron, followed by the linkage of the exons back together, creating the type of bond that you normally find in nucleic acids, the so-called phosphodiester linkage. Here's an example of the self-splicing reaction. You can see the two ends of the RNA molecule. If you follow it from the 5' to the 3' direction, the intron forms a three-dimensional shape, which allows the chemistry to occur. Those of you that are big fans of organic chemistry can follow the arrows and see how the electrons are pushed. Although it's not a complete depiction of the organic chemistry that's involved, but the end result is you end up with the intron being released and the two exons are now spliced together. Similarly, the spliceosome
ends with a very similar sort of reaction. You have an intron. It involves lots of different cofactors that collectively make up the spliceosome, and they mediate the formation of this kind of looped out intron and this sort of lariat structure, and the two exons being spliced together. So why do organisms have introns if they are just DNA that gets removed? For a long time, introns and other non-coding pieces of DNA were collectively referred to as junk DNA. As we understand more and more about DNA, we're realizing, or in the genome in general, we are realizing that a lot of the things that previously had been discounted as non-functional or junk now have functions, and introns are starting to fall into that category. One potential advantage of having introns is a phenomenon that's referred to as alternative splicing. And so alternative splicing is defined as a pre-messenger mRNA, a pre-mRNA that has multiple introns that can be spliced together in different combinations. So to look at this from a visual point of view, if we see a gene, the introns are shown in green, the exons are shown in red. You have 4 different exons, and you could, for example, have alternative splicing that occurs whereby which this exon is spliced to exon number 2, which is spliced to exon number 3, but exon number 4 is removed as part of the intron processing, and you end up with a messenger RNA that would encode protein A. Alternatively, you can have exon 1 linked to exon 2, and then these 2 introns, as well as the intervening exon 3, are spliced out, allowing exon 2 to be directly adjoined to exon 4, creating a completely different messenger RNA that would encode a different protein. Here is a depiction of how this often looks in real scenarios, where we have a fairly complicated pre-mRNA transcript that has multiple different exons. The way that they are adjoined are shown by these kind of dotted caret lines. And you can see there are a large number of variations of different ways that this pre-mRNA transcript can be spliced together, each of which making a different mature mRNA and therefore a different protein product. And you can see that they are, in this case, the way that they're spliced differs by cell type. So different types of cells, whether they're particular types of muscle cells, brain cells, fibroblasts, they all have different splicing patterns, so it allows a single gene in the genome to create different gene products in different cell types.